about 6 weeks left. We got a lot of ground to cover. So, we'll be moving kind of fast here in a little bit, particularly with the ones on energy and so forth. So we can, I want to make sure we get as many exams in before you. So stick with it and make sure you listen to the things that I'm telling you to make sure to know for the exam and so forth. That will be running out before long. Basically, once we get Thanksgiving break is almost over. Like 2 weeks maybe after that, and we're done. So it's going to be important to get all of these chapters in, but it's also going to be very important for you to make sure to do well on them. And today we are going to pick up talking about the energy. We're going to move fairly quickly on some of these things. There's going to be a lot more information in the chapter than what we are covering, so make sure you pay attention to what I tell you to stick with, okay? And today we'll pick up here with the setup on energy that we were talking about before. We talked last time about forms of energy. You should know those. We had talked about what exergonic reactions were. Exergonic gives off energy whenever the reaction takes place. Catabolic reaction. Endergonic would be an anabolic type reaction in which energy is absorbed, it's used. Endergonic reactions are going to use and store energy. Exergonic reactions are going to release and give off energy. Remember, we said cellular aspiration that we're going to be talking about is an exergonic reaction. And you can pretty much count on having those questions on the exam. Notice here we're talking about the only time, and this is something else to remember, the only time a cell reaches equilibrium or an organism is going to reach equilibrium is going to be in death. Once it reaches equilibrium, then everything in it stops and all of its reactions stop. So that occurs in death. Notice they're not in equilibrium. We're open systems and there's constantly things happening and going on. Notice this, a defining feature of life is the fact that cells never reach equilibrium. These are what we have talked about. A catabolic pathway in a cell releases free energy. Closed and open hydroelectric systems can serve as analogies. ATP. Hopefully everybody has heard of ATP, adenosine triphosphate. It is called adenosine. It's a base, a nitrogen base, and it has 3 phosphates attached to it. It's adenosine triphase, or adenosine triphosphate. If it has 2 phosphates, it's called adenosine diphosphate. If it only has 1, and there are situations where that is used in signaling and in inflammatory reactions and so forth, it's just AMP, but it is also called cyclic AMP, cyclic adenosine monophosphate, but that's going to be used in other situations. Now, these bases, this base and the phosphate with a different number of phosphates occurring for different things in different places. What we're concerned with right now is only ADP. When we have an adenosine with 2 phosphate, it is an uncharged battery. When we add the 3rd phosphate, it's charged. That 3rd phosphate, the bond is going to contain energy that can then be released elsewhere in the cell, so that energy can be used to make proteins or whatever that needs to be done, what work that needs to be done by the cell. Notice the cell itself does these three kinds of work: chemical, transport, and mechanical. That is not energy. Don't confuse that with chemical energy or mechanical energy. This is work. To do work, cells manage energy resources by energy coupling the use of an exergonic process to drive endergonic. Cell needs to use energy, for example, to make protein. To do that, it must have the energy that it gets from an external reaction, such as metabolism or cellular respiration. And notice here, what manages this energy coupling is ATP. Now, when we study photosynthesis coming up, what you're going to find, we've got an ADP sitting in the cell. Sunlight, the photons will hit chlorophyll. That sunlight, the photons will excite electrons in the chlorophyll to contain more energy than what they are allowed to have in their shell. They can actually jump to another shell, give off that energy, and fall back to where they're supposed to be, the electron. That ADP can take then that energy and use it to put another phosphate, take it to a different place in the cell, break the bond, release the energy, and then the cell can do work. That's all photosynthesis is. And primarily, the first molecule that will be made will be that CH2O, that real simple form of sugar. Then that can be transferred into glucose. It can be rearranged, used to make oils, whatever that needs to be done. So cells have to have energy. That energy is going to be transferred in the form of ATP. Adenosine triphosphate is the carrier. I like to call it a battery. If it's uncharged, it's ADP. Once it's charged with phosphate carrying energy, it's ATP. Notice, ATP is composed of ribose, adenine, and 3 phosphate groups. Remember back now, if this was only adenine and ribose, we would call that a nucleoside. If we added a single phosphate to it, a nucleotide, it can serve. If we add 2 to it, now it becomes diphosphate, and 3 is mono. So this basic form, these bases, particularly adenine, is used in a lot of different areas. And think about it, it makes sense. Why have a different molecule for each and every little reaction if you can use the same resources like adenine and sugar and do a lot of different things with it, just change the arrangement a little bit, keeps the resources down, and it's just more efficient system. You already have had to draw adenosine triphosphate. You may have to again, 3 phosphates. One thing I want to mention to you, you won't get this until you get into cell biology, but there is something that happens in this process called delocalization of electrons. In other words, they don't always go where they're supposed to go. Some of these have such a pull for it, such a draw, that particularly these first 2 can keep more of the electrons in their area. They share it, and they have actually, it's a stronger bond right here than it is between this one because of the way they deal with electrons. Delocalization. You can actually measure the amount of energy that it takes in those bonds to hold those together. ADP, therefore, is not going to be real easy to break apart. ATP is, because this bond is not as much. And as I mentioned to you before, when these electrons start to get into sync with each other, think of it like 2 speakers that are in phase. And whenever you have those 2 speakers in phase and the same thing coming out, everything sounds real clear and positive. If they are out of phase, like you get all high school gym, because of the setup, you got everything going each wave hits each other, the waves, they bump into each other, they cancel out, and everything sounds like a blurb. Whenever these work with each other, that's called being in resonance. Think of it as vibrating at the same time, the same speed together, or you have one vibrating at one speed, one at another, waves clash, and they interfere with each other, they weaken each other. When they're in resonance, they're stronger. These are in resonance. This is not in with them.
So the adenosine is basically the adenine plus the ribose, right?
It's the same nitrogen base we've been talking about with the ribose, except in this case, if we've got ADP, we've got only 2 phosphates attached. If we've got one, we've got a nucleoside. When we add the 1 phosphate, we got a nucleotide. When we add 2 phosphates, now we've got
adenosine triphosphate?
diphosphate, and 3
Oh,
we have triphosphate.
yes. Did you say that if it has 1 phosphate, it can also be called adenosine monophosphate?
It is called cyclic AMP,
Oh,
adenosine
AMP.
monophosphate. This shows whenever we get this particular situation right here, if we add a phosphate to it, we get that. Notice, what do we have to do? What kind of a reaction? A hydration reaction. We're going to make this molecule, if we were going to add, it's a dehydration reaction, right? It gets rid of a water molecule to make places on the ends so that we can have that attachment available. So we can add a phosphate to it. We have to have energy to drive that, then we get ATP. It's really just a different version of what we've already learned, not that much different. Notice the bonds between the phosphate groups can be broken by hydrolysis. What is hydrolysis? Add a water molecule to take up those ends, free nerve endings that's going to be released whenever we take those things off to meet the octet rule again. And it's obviously not nerve endings, but kind of like a live wire if we take that off. Energy is released from ATP when that terminal phosphate, the 3rd phosphate, is broken. Remember, energy in a molecule is stored in the bonds. The release of the chemical energy comes from the change from a state of low energy, not from the phosphate bonds themselves. Yeah.
Did you say that the terminal phosphate was the 3rd one? So
Yes.
that's the one closest to the ribose? Is that what you're saying?
No.
Okay, so the 3rd one on the left.
Farthest away, up here.
Okay.
Okay, the last one attached to the chain.
So between that one and the phosphorus next to it, and that's where the energy is released.
If you number these 3, 2, 1, 1 closest to the adenosine, whenever 3 separates from 2, the energy is released.
Thank you.
These 1 and 2 are in resonance, and they are tougher. You'll find when you get into cell biology.
Thank you.
Remember the term right here on the first. Bonds between the phosphate groups of the ATP's tail can be broken with hydrolysis, and then with hydrolysis you release the monomer. Remember, when you expel water, it puts 2 monomers together. Now, while these are not monomers, it's still the dehydration reaction or hydrolysis because it gets rid of the water and takes water. In other words, dehydration reactions and hydrolysis can occur on things other than water. And there are enzymes, remember, that will take a water molecule out of a bond. There are enzymes that will put one back in. That is their sole jobs. 3 types of cellular work we mentioned: mechanical, transport, and chemical. They are powered by the hydrolysis of ATP, meaning take a phosphate away, add a water molecule to it, energy is released, what we just looked at. Hydrolysis is going to release energy from that 3rd phosphate. In the cell, The energy comes from an exergonic reaction of ATP hydrolysis. When ATP hydrolysis occurs, that third phosphate is broken, then energy is given up. That energy can be used to do work then, such as make another protein. Endergonic reaction, remember, endergonic absorbs energy and can be used to go through an anabolic process such as building a bigger molecule. This gives you some examples. You do not have to know those, but it's just showing you here glutamic acid and ammonia can come together to make glutamine. The free energy of glutamine, 3.4 kilocalories per mole. Notice down here, glutamine, ammonia with ATP, and make that plus ADP plus a free phosphate, and the free energy given off 7.3 kilocalories per mole, which is the general type of reaction. That's a pretty decent average for most of these. You don't need to remember those numbers. Now, notice the terms here. This is what you'll hear later on in many different things. ATP, remember, drives an endergonic reaction. Endergonic reactions use energy. Where does that energy come from? The hydrolysis of ATP, which is an exergonic reaction. ATP drives endergonic reactions by phosphorylation, is a term that you'll need to remember, which means transferring a phosphate group to another molecule. So if we transfer a phosphate group from one molecule to another one, that molecule that receives the phosphorus is said to be phosphorylated. The recipient molecule is now called phosphorylated intermediate. Basically, all you have to remember is been phosphorylated. This shows you some of the work. A transport protein, such as one that can take a molecule from outside of the membrane and allow it to come in or go out. So that's transport work. Mechanical work. AT binds non-covalently to motor proteins and then is hydrolyzed. So it binds here. ATP comes in, binds, is broken down, ADP to the phosphate. Regeneration of ATP. One of the reasons this system works is because spread throughout your cells will be that little intermediate ADP. In other words, it's there, it's available, waiting to be phosphorylated. All it needs is the energy in the phosphate and an enzyme to drive the reaction. ATP is a renewable resource that is regenerated by the addition of a phosphate to ADP. The energy phosphorylate ADP comes from catabolic reactions in the cell. In other words, when the cell makes ATP from ADP, it's because a chemical reaction, an exergonic reaction, occurred that released energy. So now the ADP can be phosphorylated and bond and hold that energy. ATP cycle is a revolving door through which energy passes during its transfer from catabolic to anabolic. Remember, catabolic builds or breaks down big molecules and releases energy. Anabolic consumes the energy, an endergonic reaction. ATP within the cell exists only for a matter of seconds. It doesn't, it's not something that a cell will release it and you come back an hour later and it's still there. It's going to be gone 5 to 10 seconds. That ATP will move to where we're supposed to move to within the cell now. All of this is occurring. It can go from one part of the cell, release energy, and then something happens, some kind of work's done. In cells, such as muscle cells, that require a reserve of energy, that is where you will see creatine phosphate come in. Creatine phosphate can be phosphorylated and hold energy just like ATP, but it can last for a longer period of time. So in muscle cells that may need a quick push of energy, it can store creatine phosphate to be used as its energy source, then regenerate that creatine phosphate just like ATP is regenerated from ADP. and the energy source usually for creating that would still be the same thing, catabolic reactions and so forth. Yeah.
Okay, so ADP basically it goes into a catabolic reaction to form the ATP because it's a phosphorus group is added to it, which would be called phosphor. Sorry, I'm not pronouncing it.
Say it again.
Phosphorylated. So the ADP has the phosphate group added to it, which would be considered a catabolic reaction.
Endergonic.
Oh,
If it adds to it and builds and stores energy, it's endergonic. Endergonic uses energy. ADP would use the energy to phosphorylate to become ATP. Then when ATP gives off that phosphate, it is catabolized. It breaks down in an exergonic reaction that gives the energy.
Okay.
So is exergonic the same as catabolic reaction?
Exergonic is going to give off energy, yes.
Okay.
And like that's what a catabolic reaction does when you break down a big molecule into smaller ones, it's giving off energy.
So what's the difference between the two, a catabolic and an exergonic reaction?
Catabolic is referring to building a molecule. Exergonic just means a reaction that gives off energy.
So it's not considered building a molecule if you're just adding the phosphate group. That's basically just, it becomes
Carrier.
phosphorylated, right? So the one that's actually excuse me, the one that actually is has the phosphate group added to it, that's the phosphorylated one, right?
Once ADP has phosphate added, it is said to be phosphorylated.
Okay, so energy is used to build the ATP from the ADP. It's an anabolic reaction because it's using
It would
energy,
be,
right?
It's
Okay,
making the molecule bigger.
Endergonic. Okay, and then ATP can be used in exogonic reactions, right?
ATP, if it gives off energy itself, would be an exergonic reaction.
Right. Okay. Thank you.
No, the catalyst term here in chemistry, there's certain chemicals you can add to something else and make reactions happen and happen faster, and makes the product build up faster. In human reactions, in biological reactions, most of those would not occur fast enough to support life. So there has to be a catalyst added to make those reactions occur quicker and to produce more product. That is an enzyme's job. An enzyme is a protein catalyst, as you see there. Hydrolysis of sucrose by the enzyme sucrase is an example of enzyme catalyzed reaction. So sucrose is a disaccharide. If we want to break it down into fructose and glucose, an enzyme called sucrase will come along. Sucrase will then be broken apart. That's a catabolic reaction. And you will find in some of the things, like when you get into cell biology, you will do some labs, and one of the things is yeast will be given sugar. Now, yeast is a little fermenter for bread. It can produce, primarily it's its byproduct, either alcohol or carbon dioxide. Problem is, sucrose itself has to be in a monosaccharide form. It cannot ferment a disaccharide. Now, in our bodies, we have sucrase to break that down. The yeast, over time, has evolved a mechanism in which they can break down a disaccharide and ferment it like sucrose. But the way they do it, they can do that because they make their own enzyme called invertase that can then break down the sucrose into the fructose and glucose. So if you were making wine, for example, and fermenting it, you could add straight sucrose to it, and the yeast will produce that enzyme and break it down into the individual monosaccharide constituents and be able to ferment it. If you were, on the other hand, going to use a complex carbohydrate like corn that's made from where we get ethanol from through fermentation, don't work that way. That yeast can't break down that complex carbohydrate. So what you have to do is you either have to get that enzyme in there one way or another. Either you put it in, or you put something in that's got that enzyme. The easiest way to do that, it's called mashing. When you hear the term mash, made like with ethanol production, whether it's the kind you drink, ethyl alcohol, or the kind that goes into gasoline, that is done by getting the corn to start to sprout. Just as soon as it starts to germinate, then the enzymes, it releases its own natural enzymes that will start to break down that complex carbohydrate into the individual sugars, and then the yeast can work on it. So that's why when you hear that term mashing of corn, that's what's happening in there. But it all comes down to these little enzymes. Sucrose, notice C12H22O11. Add some water to it. Why are we going to add water?
Because it will break the bonds between the disaccharide. It'll break
It's
the
hydrolysis reaction, so we have to add water to take off these free ends that's not being accounted for with the octet rule to make it stable. So we add water, and then you come down with the glucose and fructose. Every chemical reaction between molecules, I'm just going to summarize this. For any chemical reaction to take place, there is a certain amount of energy required for that reaction to take place. It drives the reaction. It's the force that causes the reaction to take place. That is called the activation energy. The initial energy needed to start a chemical reaction, called the free energy of activation, or activation energy. Remember, that has to be free energy, energy that is available. Activation energy is often supplied in the form of thermal energy that the reactant molecules absorb from their surroundings. Think about something. If you've ever tried to lose weight, you probably find it difficult, like most people. But yet you can get the flu and go for about a week, and you can lose a lot of weight real quick. What do you think's happening? How come you can get sick with the flu and lose weight at 3 times the speed or more?
Because your body has to use energy to fight the virus, therefore you...
Your metabolism has to increase your cellular respiration. You're not taking any energy in. The reason you don't want to eat when you are sick, your body makes you that way because your body needs that blood and all of its energy to be fighting those microbes. It does not want to use that blood supply to be digesting food. It's a waste to it. It's got more important things it needs to do. And because the amount of energy your body is using goes up enormously because you're fighting that. Remember, often associated with these things are fever. What's fever? That residual heat right there from these reactions that are going on that your body gives off because it's going through metabolism so much faster, more. So your body is using a lot of energy, and you'll lose weight much faster when you're sick like that because your body just kicks in and you're not taking in food. And you get nauseated simply because your body don't want you putting junk down in your stomach that it has to digest. It wants that blood to be spread throughout your immune system fighting. It doesn't want to be all isolated. When you eat a large meal, you oftentimes get sleepy. The reason is when you eat a large meal, it takes an enormous amount of blood to break all that down and to carry the parts to the different parts of the body. So the blood pools in your abdomen area to go through digestion. Because the blood is pooling in your abdomen area, there's not enough blood up into your brain, you're not getting oxygen, so you get sleepy. After you eat a big meal, you just want to sit. You want to get in an easy chair, take a nap. You don't want to be out moving around because even though you just have eaten, you don't have any energy for that. It's all down here digesting and doing work. Enzymes. Remember, every reaction takes a certain amount of energy in order for that reaction to occur. Enzymes lower the activation energy. Think of it this way. If we've got two chemicals, two reactants right here, and they are going to come together to make a product, it's gonna take a certain amount of energy to bring them together. Enzyme does is put them like that. They come together that far instead of that far. Enzymes have a place where it can hold both reactions and put them together, so when the reaction starts, wham, they're there. So it takes less energy. Enzymes, remember, low activation energy, and I will probably ask you that on the exams. How does an enzyme speed up a reaction? The answer, by lowering the activation energy. Note, enzymes do not affect the change in free energy. Instead, they hasten the reactions that would eventually occur. They just make it happen faster. The reactant that an enzyme acts on is called the substrate. The enzyme binds to its substrate, forming an enzyme substrate complex. An enzyme by itself can do nothing. It has to have something to act upon. What it acts upon is called the substrate. On an enzyme, there will be a particular spot, a particular conformation. Remember, we said a protein's function is determined by its shape. There will be a particular shape on that enzyme that will match perfectly with its substrate so that it can hold that substrate. That is called the active site on the enzyme. Induced fit of the substrate brings chemical groups to the active site. That means the substrate fits the enzyme's active site. That's what that induced fit is. Like a key in a lock. It brings them together close. It's a good fit. Notice right here, our substrate. Here's the enzyme. And remember, all of these little blue things you see are going to be the atoms and molecules that make up amino acids that come together to make that protein, which the enzyme is a protein. It's got its bonds, disulfide bridges, and hydrogen bonds that keep that particular shape going right there. Substrate comes along. This is the site where that will fit as you see. Now, one of the things that usually does happen too is once that substrate comes, notice this is a little bit open. Once that substrate hits, this induced fit means the enzyme actually closes around it and fits even better. One of the things that's like this that is something we do commonly. If you've got, I know you guys are probably all big time cooks, right? You go home every day and cook dinner for your whole family. I like to cook. If you're going to thicken a sauce, make gravy, whatever, flour has what? Got a lot of protein in it, glutens. Now, when you put flour in water, just flour and water, right? Nothing happens. What happens when you heat it? What does heat do to a protein? Denatures it. It takes all of this, instead of being in that wad, it brings it out into big long strings. When you do that with flour and a liquid that's got, like, say, beef broth or something in, or if you're using starch to do the same thing, whenever that denatures, it will start then to curl back and want to come back and reshape itself. But this time it's got junk in the way, like all that beef broth, and it will enclose around them, and that's where the thickening comes from. You've got these protein strands through there that's holding clumps and whatever it is you're thickening, and that's the way things like starch or whatever thickens a sauce. Same process. What happens when you cook an egg?
It denatures the
When you
protein
cook
in
an
the
egg,
oven.
you're denaturing the proteins in it, and then whenever they don't curl back, they just stay where they're at. They don't reform. So you've got these strands of protein just overlap, and they make the yolk and the white. Then comes that solid mass instead of the liquid that they were before, because they were denatured.
You said that when the protein's in the flour, are denatured, it releases. Well, they break
It
it down.
straightens them out. Goes back to the primary form.
Ah.
Now, they will try, sometimes when you do an enzyme, they will try to reform again. Some don't, like the egg, some will. And when they try to reform, they never reform the correct way, but they incorporate whatever else you've got in that mix, and they surround it, make little pockets where that isn't trapped, and that's where the thickening comes in. See how much you can learn, chemistry, folks? You can cook now, all of it. You know what, one of the best desserts going, has been around in history forever, custard. You know what it takes to make custard?
Yolks.
Milk and eggs. You mix them up real good, and then you heat them slowly. It denatures the proteins, and then the proteins in both try to get back together. They can't. Milk's got the egg involved, and then you get this little pudding-like consistency custard. That's all it is. Lemon curd. Lemon curd, you take eggs, you whip them up under low heat and start to drizzle butter in with them. The butter gets incorporated into the proteins of the egg as it tries to reconstitute itself. And then when you add lemon juice with the eggs and stuff, you have got just like eating the peel of a lemon curd. That's all it is. Put those into little toasted cupcake-like filling. You got it made.
I do have a question. So we will need to remember how to draw adenosine triphosphate, but more complex now. Basically, adenosine with
No,
broken down
just
into
that you don't
adenine.
have to really do anything different.
So you mean just so
You
we
don't
don't?
have to draw the ribose or anything. If I ask you to draw it, it'll be the same way.
Oh, okay. Thank you.
Just as long as you know that there's a ribose there. Now, the active site can lower activation energy by these methods. Orienting, you do need to remember them. I'll probably ask you what are the 4 methods that an active site can lower the activation energy. Orienting the substrates correctly, straining substrate bonds, providing a favorable microenvironment, and covalently bonding to the substrate. Covalently bonding means it can hold them tight into position. Providing a favorable microenvironment, it surrounds them, prevents anything else from interfering with the reaction. It gives them a good, clean environment in which to react. Straining substrate bonds, that means it can pull on that substrate. Orienting the substrate so they're in the proper position. Notice up here, this is the enzyme, this is the active site. We've got 2 substrates that when those substrates fit together the way they're supposed to within this enzyme, now these substrates are held together in the proper position. So we can make a new molecule.
So is the enzyme actually going and grabbing the substrates?
It actually provides a physical place where they can be held together so that oriented correctly and come together, right?
But are the substrates doing that on their own, or is the enzyme the one that's actually going and grabbing the substrates and doing it? How you were saying it kind of like brings them together.
This is usually, the substrates and so forth are going to be provided in the environment, and they will come together by the forces like the enzymes or whatever you understand. And notice here, we've got these 2 individual substrates, and now we've got a product. Once it holds them in a position, then bonds and reactions will take place. We've got a new molecule now. So we've got different products made. Think of that enzyme as a place where things can happen fast. Enzymes can be affected by the environment, temperature and pH primarily. Remember, they are proteins, so as proteins, they are going to be susceptible to the pH of the environment and to the temperature. There are some enzymes that will only act in a range, a very small range of pH or temperature. In other words, if the temperature is too cold, they won't be active. If it's too hot, they won't be active. It has to be correct. And that enables, as a control mechanism to make sure the enzymes work when and where they're supposed to. So, enzymes can be infected or affected by chemicals, temperature, and pH. And as we said, each enzyme has its optimal temperature. It has an optimal pH. It has optimal conditions in which to operate. And cofactor are important. These help the enzyme to like a light switch, off or on. I want you to know what they can be or be made of. A cofactor may be inorganic, such as an element, or it can be organic. It could be a vitamin. So cofactors can be organic or inorganic. They could be a metal or they could be a molecule like a vitamin. Your body has a need to be able to control the action of an enzyme to make it work or to make it stop. Competitive inhibitors bind to the active site of an enzyme competing with the substrate. So let's say you've got an enzyme that's doing too much of what it's supposed to be doing. Your body can produce another little substrate that's not like the real one, but will bond to the site and block it so the one that's coming in can't get to it. So you can't make too much product. You can do that with medicines. If you've got some diseases that are controlled or affected by enzymes, and there's very few that are not, if you can shut down an enzyme, that medicine acts as a block. It bonds to the enzyme so the substrate can't, so you don't get too much of the bad product produced. There are, ever heard of things, blood pressure medicine, MAO inhibitors. They can go and block the little receptors on the blood vessel so that they won't shrink down and constrict and make your blood pressure go up. If you got some that's doing that too much, you can put a medicine in there that will block that. That's an inhibitor.
Something like an alpha blocker or something?
Beta blockers,
Oh, yeah, beta blockers.
all of it working on the same principle.
Okay.
Not necessarily an enzyme, but it's working on the same. Non-competitive inhibitors bind to another part of the enzyme, causing the enzyme to change shape. If you put the wrong cofactor with that enzyme, it can actually make the enzyme change shape so that it won't work. Now look here. Some examples of inhibitors include toxins, poisons, pesticides, even antibiotics. When these get in your system, they can prevent enzymes that your body needs to perform a function from occurring, and that's why they do damage. Notice, this is what is supposed to be happening. You've got a substrate in your body, this enzyme is supposed to work on it. Competitive inhibition, if we put something in here to block that from getting in, then the substrate can't get in, so the product will never be made. Over here, non-competitive, this is the cofactor area right here, where the cofactor comes in and attaches, then it makes the enzyme active to its job. If you put something in there, notice how it changed the shape now, so your substrate won't fit. So this is non-competitive. It doesn't compete with the substrate, it just changes the shape so it won't fit. This is competitive, puts something in its place. And remember, folks, there's hardly anything in your body reaction that takes place that does not involve these enzymes. So it's almost everything they're involved with.
Did you,
Rick?
I'm sorry, did you say that the cofactor can bind into that other side over there and make it work?
It will if it's the correct cofactor.
Oh,
Some medicines can be a cofactor that will change the shape, so the substrate won't fit. There is one, it might be an element, or it could be a vitamin that will attach to it and then turn it on. If you put something in place so it can't get there, then once again, it won't work.
thank you.
Now, notice it says, chemical chaos would result if the cell's metabolic pathways were not regulated. A cell does this by switching on or off the genes that code specific enzymes, or by regulating the enzyme. Sometimes you can tell the gene to stop making enzymes. There's something like this that happens in alcoholism. One of the reasons that people become alcoholic is you've got a certain chemical that controls your metabolism, turns it off and on. In some people who become alcoholic, the alcohol itself takes the place of that enzyme. It does the same thing that the enzyme does as far as controlling metabolism. After a while, if someone's an alcoholic, your body's got all the alcohol in it, it doesn't need to produce that enzyme, so it stops making it. So what happens? If you stop drinking then, you don't have the enzyme for metabolism, you'll start to get those cravings. Now your body's starting to rely on that alcohol to take the place of the enzyme. And what do you notice about most alcoholics? They'll have a real red face and things. They're producing a lot of heat. Their metabolism really kicks up because of that alcohol. So that's an example where something like alcohol can act as, doesn't act as an inhibitor. It just tells the gene stop making the enzyme that we need. Allosteric regulation may either inhibit or stimulate the activity. Notice, it occurs when a regulatory molecule binds to a protein at one site and affects the proteins at another. Most of these allosteric enzymes are regulated are polypeptide units. Notice we're not calling these proteins now, they're just a string of amino acids. Each enzyme has active and inactive forms. The enzyme itself, the binding of an activator can turn the enzyme on and make it function. The binding of an inhibitor can turn it off. Don't worry too much about cooperativity right there. Don't really worry about the identification of these regulators here. If you look right here, it will show you some of the things that can happen. This is just not that much different than what we were talking about before. I want to ask you about these particular ones. Feedback inhibition is one I do want you to know. In other words, there's supposed to be a product made by these enzymes after it goes and does its work. Once that product gets in your system, the fact that it is present tells the enzyme don't make any more. In other words, the presence of the product turns it off. That is called feedback. Notice, feedback inhibition prevents a cell from wasting chemical resources making more products than what it needs. Here's another example of it. We will stop there. Running out of time. That is the last slide. So we'll look at that last one next time, and then we will get into the big daddy, cellular respiration.
When do you expect to test? Do you know yet?